Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 45. The topic is effects of local conditions on enzyme activity, cofactors, and enzyme inhibitors. Discussing agenda for the topic will be effects of local conditions on enzyme activity, effects of temperature and pH, cofactors, and enzyme inhibitors. Lecture outcome. After watching and listening this lecture, students will know about the various factors affecting enzyme activity. Introduction. The activity of an enzyme, how efficiently the enzyme functions, is affected by general environmental factors such as temperature and pH. Obviously, enzymes function inside a living system, so these environmental factors, that is temperature and pH, refers to the environment inside the living system. It can also be affected by chemicals that specifically influence that enzyme. So enzymes are not only under the effect of temperature and pH, but also chemicals. In fact, researchers have learned much more about enzyme function by employing such chemicals, just like in molecular biology we have a technique called gene knockout technique. It is used to know about the function of a particular gene. If a particular sequence of DNA is not known in terms of function, then mutations are induced in that sequence and the individual or the animal is looked for showing any symptoms. Similar is the case, the enzyme activity or the function of a particular enzyme is found by blocking this enzyme, blocking the active site of an enzyme, and then looking for any symptoms which can be attributed to this enzyme. The three-dimensional structure of proteins are sensitive to their environment. Once again, I am repeating the phrase structure determines the function, and this is important for proteins. Their three-dimensional structure is vital, is the key to the function they perform, but this three-dimensional structure is vulnerable to deteriorations in the environmental factors. As a consequence, each enzyme works better under some conditions than others. So there is a specific optimal conditions under which the enzymes show its most efficient activity. Because these optimal conditions favors the most active shape of the enzyme, because these optimal conditions does not deteriorate the three-dimensional structure of the enzyme. Temperature and pH are environmental factors important in the activity of an enzyme. On the right side of your screen, you can see this is a normal protein perfectly molded into its three-dimensional structure, and upon denaturation, the structure is completely distorted, and as a result, now this protein is unable to perform the function for which it was produced. and this process can go in reverse direction, and when the conditions become optimal, then the three-dimensional structure is restored. Up to a point, the rate of an enzymatic reaction increases with increase in temperature. Now, once the temperature starts increasing, initially it has a positive effect on the ability of an enzyme to catalyze reactions. And what is the reason why temperature has positive effect initially? It is partly because substrate molecules collide with active site more frequently when the molecules move rapidly. So when the temperature increases, the substrate molecules are excited, and there is more probability that they will collide with the active sites of the enzyme, and the products will be formed. Above that temperature, however, the speed of the enzymatic reaction drops sharply. Now, initially, the temperature helped the substrate molecules to come into an excited state, but when temperature increases beyond this limit, then what happens? Then the problem occurs in the three-dimensional structure of the enzyme. The thermal agitation of the enzyme molecule disrupts the hydrogen bonds, ionic bonds, and other weak interactions that stabilize the active shape of the enzyme. Now, up to a certain limit, the temperature increase was directly proportional to the rise in the enzyme's activity, but beyond that point, the enzyme was unable to hold on to its correct conformation because of the breakage of bonds, disruption of bonds, and as a consequence, the three-dimensional structure of the enzyme distorted, and finally the enzyme molecule denatures. Each enzyme has an optimal temperature at which its reaction rate is greatest. Now there are optimal levels for each enzyme. Without denaturing the enzyme, this temperature allows the greatest number of molecular collisions and the fastest conversion of reactants to product molecules. So if the temperature is not at a point when it denatures the enzyme, so below that point the temperature increases the collision of the molecules and thus the conversion of the reactants into products, but there is a specific high limit, and beyond that point then this increase fires backwards and it deteriorates the enzyme's shape. Most human enzymes have optimal temperature of about 35 to 40 degrees centigrade. So this is the temperature range under which most of the enzymes found in the humans function at their peak levels. And this 35 to 40 degrees centigrade lies within the range of the normal body temperature. However, there are certain exceptional cases where the enzymes can tolerate extreme temperatures. One of the examples is the bacteria which are found in the Yellowstone National Park in America. These bacteria can survive in boiling waters. Thermus aquaticus is one of the examples. The enzyme which replicates the DNA of this bacteria is used in PCR to replicate the DNA, because PCR has 3 stages. The first is denaturation, the second is annealing, the third is polymerization. So the ordinary enzyme is unable to withstand the denaturation temperature, and the enzyme is also denatured. So the discovery of Thermus aquaticus bacteria and the enzyme in that bacteria was one of the major boosts to molecular biology, because the enzyme was used in PCR, in vitro DNA replication. Now here is a comparison of the 2 enzymes. One is found in the humans, the other is found in bacteria, in thermophilic bacteria, which lives in extreme environments. Remember, from the beginning of this course, we studied archaea, which lives in extreme environments. So cyanobacteria thriving in the hot water of Nevada geyser. Now, this is the enzyme found in the human body, and its peak activity, this is the rate of the reaction. So the rate of the reaction is at its peak at 37 degrees centigrade, and when the temperature increases beyond this point, then the rate of the reaction drops considerably. On the other hand, the optimal temperature for the enzyme found in this thermophilic bacteria is almost 75 degrees. So there is a good comparison that the optimal ranges of enzymes also vary. Just as each enzyme has an optimal temperature, it also has a pH at which it is most active. The optimal pH values for most enzymes fall in a range of 6 to 8, but there are exceptions. Now this is the optimal range, 6 to 8. But here is an astonishing example of the pepsin enzyme, which is found in the stomach, because the stomach has highly acidic environment courtesy of the presence of hydrochloric acid. So pepsin works best at pH of 2, a highly acidic pH. On the other hand, trypsin functions at pH 8. Trypsin is there in the intestine, and intestine has alkaline environment, so the optimal pH for trypsin is 8 and the optimal pH for pepsin is 2, because these 2 enzymes are found in different localities in the body. Therefore their pH ranges, normal pH ranges, are also different. For example, pepsin, a digestive enzyme in the human stomach, works best at a very low pH. Low pH means acidic pH. Such an acidic environment denatures most enzymes, but pepsin is well adapted evolutionarily to maintain its functional 3D structure in the acidic environment of the stomach. In contrast, trypsin, a digestive enzyme residing in a more alkaline environment of the human intestine, would be denatured in that stomach. So if you take pepsin enzyme and put it in the intestine, it won't function there, because its optimal range is 2 and in intestine it's 8. Same is the case with the trypsin. Trypsin will not function in the stomach because the pH of the stomach is highly acidic. Now, cofactors. Many enzymes require non-protein helpers for catalytic activity, often for chemical processes like electron transfer, that cannot easily be carried out by the amino acid in proteins. The amino acids lining the active site, they interact with the substrate molecules. The R groups of the enzyme, the R groups of the amino acids lining the active site interact with the molecules of the substrate, and this interaction destabilizes the bonding in the substrate molecule. Sometimes the amino acids present in the active area are unable to do so, therefore they need a cooperation from other helpers, other non-protein helpers. These adjuncts called cofactors may be bound tightly to the enzyme as permanent residents, or they may bind loosely and reversibly along with the substrate. So the cofactors, they either bind with the enzyme as permanent residents, or they bind loosely and reversibly with the substrates. That means they bind with substrate for a certain period of time, and once the catalytic reaction is over, then they are detached. The cofactors of some enzymes are inorganic, such as the metal ions, metal atoms, zinc, iron, and copper in ionic form. Now, if the cofactor is an organic molecule, then this cofactor is referred as coenzyme. If the cofactor is an organic molecule, then it is referred to as coenzyme. Most vitamins are important in nutrition because they act as coenzymes or raw materials from which enzymes are made. So the vitamins, which are micronutrients, which are required in minute concentrations to the body, for which purpose? Because they act as coenzymes, they are organic and they act as coenzymes, or they may be the precursors from which coenzymes are made. Now, enzyme inhibitors, certain chemicals selectively inhibit the action of specific enzymes. They hamper the activity of the enzymes. Sometimes the inhibitor attaches to the enzyme by covalent bonds, in which case the inhibition is usually irreversible. So when the covalent, the interaction via covalent bond is established between the inhibitor and the enzyme, this inhibition is usually irreversible, means the enzyme is blocked forever. Many enzyme inhibitors, however, bind to the enzyme by weak interactions, and when this occurs, the inhibition is reversible. So when the interaction between the inhibitor and the enzyme is through a covalent bond, then usually this type of interaction is a permanent one. However, when the interaction between the inhibitor and the enzyme is through weak interactions, then this type of interaction can be reversed and the enzyme can return back to its active form. Some reversible inhibitors resemble a normal substrate molecule and compete for admission into the active site. Now, active site is that specific site in the enzyme where the substrate binds. So sometimes the inhibitor competes with the substrate for the active site. Obviously, when the substrate binds to the active site, the enzyme will catalyze the reaction. When the inhibitor binds to here at the active site, then obviously in that condition the enzyme cannot bind the substrate, and therefore this enzyme will become inactive. So there is competitive inhibition. This is when the enzyme and the inhibitor compete for the active site. This type of inhibition is called competitive inhibition because there is a competition between the substrate and the inhibitor to bind to the active site. These mimics, called competitive inhibitors, reduce the productivity of enzymes by blocking substrates from entering active sites. So once the inhibitor is attached here, then the enzyme can no longer catalyze a reaction because it cannot bind substrate in the correct orientation. Now another type of inhibition is non-competitive inhibition. In non-competitive inhibition, the inhibitor does not compete with the substrate because the point of attachment of the inhibitor is not the active site but another site which is called the allosteric site. So when the inhibitor binds to the allosteric site, it causes a change in the conformation or the structure of the active site, and then this active site does not bind the substrate correctly. This kind of inhibition can be overcome by increasing the concentration of substrate so that as active sites become available, more substrate molecules than inhibitor. They are talking in this paragraph about competitive inhibition. So how you prevent a competitive inhibition? Increase the substrate molecules. When substrate molecules are in higher concentration, then obviously there will be more probabilities of binding of substrate molecules with the active site of the enzymes. In contrast, non-competitive inhibitors do not directly compete with the substrate to bind to the enzyme at the active site. Now, non-competitive inhibitors, just like the name suggests, it doesn't compete with the substrate for the active site of the enzyme. It actually attaches to another site present in the enzyme, which is called the allosteric site. Instead, they impede enzymatic activity by binding to another part of the enzyme. The interaction causes the enzyme molecule to change its shape in such a way that the active site becomes much less effective at catalyzing the conversion of substrate to product. So in non-competitive inhibition, the inhibitor molecule attaches with the enzyme through its allosteric site, and once the inhibitor attaches at the allosteric site, it causes a change in the conformation in the structure of the active site, and this active site is no longer able to bind the substrate correctly. Toxins and poisons are often irreversible enzyme inhibitors. Toxins and poisons, once they get themselves attached with the enzymes, then this reaction cannot be reversed. One of the examples is sarin. Sarin is a nerve gas, and this gas was released in one of the Middle East countries, and it resulted in the martyrdom of 100 people and injuring 100 more. Now, what was the effect of sarin on the molecular level? These small molecules bind covalently to the R group on the amino acid serine. Serine is an amino acid, so sarin gas binds to the R group of serine amino acid, which is found in the active site of the acetylcholinesterase. This is an enzyme which is responsible for the breakdown of acetylcholine. Acetylcholine is a neurotransmitter, and after conveying the message, the acetylcholine must be broken down, otherwise its presence is harmful for the individual. So this enzyme breaks down acetylcholine. Now what serine gas do? Serine blocks the R group of the serine amino acid, and once that happens, the acetylcholinesterase can no longer break down acetylcholine. Other examples include the pesticide DDT and parathion. These are the two pesticides which are responsible for the elimination of the pests which feed upon the crops. Now they also inhibit key enzymes in the nervous system of pests. The farmers usually spray their crops to avoid damage from insects or pests. So these are the most widely used pesticides, and their mode of action is that they inhibit key enzymes in the nervous system of the insects, and as a result the insects dies away. Finally, many antibiotics are inhibitors of specific enzymes in bacteria. Now bacteria, majority of the bacteria are pathogenic to mankind. They cause a lot of diseases, and mankind has developed ways and means, the armory, to minimize the pathogenic effects of bacteria, and one of the potent drugs is antibiotics, which is used against bacteria. Now the mode of action of antibiotic is similar to an inhibitor. Inhibitor blocks important enzymes. Similarly, the antibiotics also blocks certain important enzymes in bacteria, and bacteria is no longer able to synthesize its cell wall or produce pathogenic proteins. For instance, penicillin. Now penicillin is one of the first antibiotics to be discovered. It was discovered by Alexander Fleming back in 1928, and then in 1940s its commercial production started. What is the mode of action of penicillin? It blocks the active site of an enzyme that many bacteria uses to make cell walls. Bacteria cell wall is made up of peptidoglycan, and obviously bacteria have enzymes to synthesize cell wall for them. So this antibiotic penicillin, it blocks this particular enzyme, so bacteria is unable to make cell wall and becomes vulnerable. Citing enzyme inhibitors that are metabolic poisons may give the impression that enzyme inhibition is generally abnormal and harmful. In fact, molecules naturally present in the cell often regulate enzyme activity by acting as inhibitors. Now, the inhibitors are present inside the body, but these inhibitors, they binds to the enzymes only for a short period of time, and this process is beneficial because it helps the cell to regulate its activities. However, if the inhibitors are from an outside origin, then quite often they are harmful. Such regulation, selective inhibition, is essential to the control of cellular metabolism. Now suppose an enzyme is catalyzing a reaction, it is forming a product, and now the product formed by this enzyme is in surplus amount, which is more than required by the cell. Now there is a need that this particular enzyme is inhibited or blocked from making more product because the product is already in surplus amount. So these type of interactions which are in vivo in origin are beneficial for the cell and the organism. This was the end of the lecture. Thank you.